Hello everyone, this is Dr. Madhusree Chatterjee and welcome to my channel MC Biology Classes. This is my second lecture video and this is not for my school kids but for my senior kids. Yes, this lecture is on immunology for GATE Biotechnology aspirants. Okay, but that doesn't mean others can't watch this video. And this is the first part of this lecture. If you like this video, do not forget to give it a big thumbs up and share it with your friends. Those who are new to my channel, please do subscribe it for many more useful and interesting lectures. So without wasting any more time, let's get started. Here I have given the complete syllabus of immunology, which is included in GATE Biotechnology syllabus. The complete syllabus has been provided here, and I will start from the very beginning of this syllabus. Now, the first question that comes is, what is immunology? Before giving answer to this question, let me tell you what is immunity. In today's scenario, when the total world is suffering from a global pandemic, the term immunity has become very common to all of you. We all have become very much familiar with this term, right? Now, immunity is the ability of an organism to resist infection by pathogens or state of protection from infectious diseases. Now we get to hear from different person that his immunity is very less, that person's immunity is very high. So it means that the ability of a person to resist infection varies and it is dependent on a number of factors like pregnant ladies, little kids, then old aged persons, people on steroid medicine, or persons suffering from some chronic infections. They all are known to have lesser immunity than healthy normal individuals. Okay, so lesser immunity means their ability to resist infection is much less. So this is immunity. Then what is immunology? Immunology is the science that is concerned with immune response to foreign challenges. Okay, now what do we mean by foreign challenges? Foreign challenges means whatever that is not a part of our body is foreign to us. It may be a bacteria, it may be virus, it may be any fungus, then protozoa, helminth. All these are foreign to our body, right? And what we call these foreign challenges? Yes, they are known as antigens. Okay, foreign substances are known as antigens. So immunology is the science that is concerned with immune response to foreign challenges. The term immunity was derived from a Latin word immunis, which means exempt. And the last term that I have mentioned here is immune system. Let us consider our body as a big castle or very big palace where normally gatekeepers, security guards, watchmen are present in order to protect that castle from protrusion, from intruders. From intruders, they need to protect the big castle. So if we consider our immune system as our security guard, it consists of cells and tissues and organs, and they carry out the function of protecting our body. So immune system consists of array of different cells, tissues and organs to give us protection against infection by pathogens, right? Now, history of immunology. Okay, don't get afraid. I'm not getting into the detail of history of immunology. Normally, very few questions come from this particular part of this chapter, but I'll mention only those parts that are important in order to study this chapter. Okay. The advent of immunology began in 430 BC. Peloponnesian War, Thucydides described plague. We all know that plague is a deadly disease, right? Okay, I am asking you one question. Just write it in the comment section if you know the answer. Name the causative organism of the fatal disease plague. Okay, name the causative organism of plague. You please mention it in the comment section if you know the answer. Okay, let's start. So in 430 BC, Thucydides described plague that ones who have recovered from this disease could nurse the sick without getting the disease a second time. That means the persons who were affected by plague already developed immunity against this particular disease, so they can nurse the sick patients. Then in 15th century, Chinese and Turks used dried crust of smallpox, either inhaled into nostril or inserted into small cuts in the skin. The technique is called variolation. And then finally in the year 1798, the landmark invention, Edward Jenner developed smallpox vaccine. There is a small story behind this. Edward Jenner noticed that the milkmaids that contracted cowpox is a mild disease, were immune to smallpox, which is a fatal disease. So he started thinking, what might be the reason behind this? To confirm his idea, what he did was he inoculated an 8-year-old boy with a fluid from the cowpox pustule, and after that, he intentionally exposed the boy to smallpox. And as predicted, the child did not develop smallpox. That means the child was already protected and the child developed immunity against smallpox. And from this whole event, vaccines came. The name vaccine was given in honor of Jenner's work with cowpox inoculation. And the term vaccine comes from a Latin word called vacca, V-A-C-C-A, vacca, that means cow. Okay. Now, here I have given you a list of Nobel Prize awardee scientists and along with their country and contribution. See, there is a tendency in GATE examination that what happens normally in GATE exam, they give column matching kind of questions. If you go through previous year question papers, you will definitely find at least 1 or 2 column matching questions in each and every year GATE question paper. So from this part, column matching kind of question might come. Like in one column, they will give the name of the scientists, and in the another column, they will give their contribution. Okay, and you have to match both the columns. So in order to do that, you need to go through the whole chart and you need to memorize the names of the scientists along with their contribution. See here, all the scientists are not much important. Like, let me tell you the most important names, like Robert Koch, you need to remember. Then Elie Mechnikov, Paul Ehrlich, then Karl Landsteiner. Karl Landsteiner discovered human blood grouping. You have studied in your, I think, 9th or 10th standard, right? Okay, then Macfarlane Barnett, Rodney Porter. Rodney Porter is very important because he is the person who gave the chemical structure of antibodies. And we will study the structure of antibodies in detail after 2 or 3 lectures. Then Rosalind Yellow, development of radioimmune assay, Caesar Milstein, Susumu Tonegawa. So these names are very important, but I would suggest to memorize all these names, okay? And here I have mentioned 2 more, okay, in most recent year, that is 2018. James P. Allison and Tosuku Honjo. Many of you must be knowing that it's very recent, right? 2018. They were awarded Nobel Prize in the field of immunology for their discovery of cancer therapy by inhibition of negative immune regulation. I would suggest the students who are going to appear in GATE exam in 2021 or 22, they should try to memorize these names, okay? Because see, even if this kind of question may not come in your GATE theory examination, but after qualifying GATE, when you will be appearing for MTech interview in IITs or some other institutes, they might ask this kind of question. You might be asked this kind of questions in your interview, because normally there is a tendency of the interviewer that they are not only interested to judge your bookish knowledge, because you have already qualified GATE exam, right? So they are very often much more interested to judge your general knowledge. So please, please, please don't forget to go through this slide, okay? Then innate and adaptive immunity. Now the actually important part starts. I would request you all to watch this video till the end, okay? Because from this slide, the most important parts are starting. Now, immunity can be divided into two components. One is innate immunity, another one is adaptive immunity. Now, what do you mean by innate immunity? Innate immunity is also known as native or natural immunity. This is inborn immunity. That means this immunity is present since birth. A newborn child also possesses this kind of immunity. Now, this immunity provides first line of defense during critical period just after the host's exposure to pathogen. Okay, I mean to say, just a pathogen has infected a host, immediately innate immunity will start acting. So it will try to provide the first line of defense during critical period. Okay. Normally, most of the microorganisms that is encountered by a healthy individual are readily cleared within few days by this system. Okay, it normally happens in case of healthy individual. I'm not talking about immunocompromised persons, but in case of a normal healthy individual, it's readily cleared. And innate immunity is less specific, without any memory. Now, what do we mean by less specific? Less specific means innate immune system is not specific to a particular pathogen, but it includes few cells and molecules that can recognize classes of molecules peculiar to frequently encountered pathogens. Okay, so it is not specific to a particular pathogen. It gives protection against each and every kind of pathogen, but it does not have any memory. Okay, so these are the features of innate immune system. Now, in comparison to that, adaptive immunity poses some different features. Adaptive immunity normally doesn't come into play unless there is an antigenic challenge to the organism. When the innate immunity fails, then comes adaptive immunity. Okay, it has a very high degree of specificity with remarkable memory. That is, what do you mean by memory? Exposure to the same antigen sometimes in future results in a memory response. And since it has memory, its response to the second challenge is quicker, stronger, and more effective in neutralizing and clearing the pathogen. Like us, when we face any challenge for the first time, it takes a longer time for us to solve it, right? But when we face it for the second time, we can respond to it quickly, strongly, and in a more effective way. Okay. So here I have given comparative analysis between innate and adaptive immunity. This is also very important for you. See, innate immunity, as I have already mentioned, responds rapidly that is within hours. It has less specificity, no memory, and it is found in vertebrates and invertebrates. Cells involved in innate immunity are phagocytes. Phagocytes include PMNs, that is polymorphonuclear cells, and macrophages. Okay, let me ask you a question. If you know the answer, please write it in the comment section. What are PMN? Okay, what are polymorphonuclear cells? I want answer from each one of you who is watching my video. Okay, so other than phagocytes, natural killer cells, mast cells, and dendritic cells are also part of innate immunity. Don't worry, we will discuss each and every type of cell involved in innate immunity in our next video, because that is going to be our next topic. Okay, and innate immunity includes few molecules: cytokines, complement, and acute phase proteins. Now just remember these names: cytokine, complement, acute phase proteins. I will describe this in my next video. Then adaptive immunity. Adaptive immunity, as I have already mentioned, it's slow to start because until and unless there is antigenic challenge, it doesn't come into action. So it takes days. It's highly specific, it has memory, and it is found in jawed vertebrates. Now the cells of adaptive immunity are T and B lymphocytes and antigen presenting cells, and molecules are antibodies and cytokines. Have you noticed something? Cytokines were a part of innate immunity. See, as well as it's a part of adaptive immunity. Okay. So from there, from this particular slide, we can easily say that innate and adaptive immunity is very much interconnected. Okay, they are highly orchestrated actually. They are dependent on each other. We will get into the details later on. Now let's start with the summary of non-specific host defenses. As I have already mentioned, innate immunity is not specific. It is not specific to a particular pathogen, right? So here we have summarized different kinds of non-specific host defenses. And here I should mention that this chart has been taken from QB, the Bible of immunology, what we can say. Please don't forget to go through QB, okay? Because QB is an excellent book. It will help you to clear your concepts very well, okay? Now let's start. First is anatomical barrier. Anatomical barrier includes skin and mucous membrane. Now, if we talk about skin, skin contains two layers. The outer layer is called epidermis, which is thinner, and inner layer is dermis, which is comparatively thicker. Now, epidermis contains several layers of tightly packed epithelial cells, and it consists of dead cells which is filled with waterproofing protein called keratin. We know that keratin is present in our skin as well as our hair, our nails, etc. Now the inner layer, that is dermis. Dermis contains connective tissue, certain blood vessels, and sebaceous glands as well as sweat glands. Okay, this is very important. Dermis contains sebaceous gland and sweat gland. Now sebaceous gland produce an oily secretion called sebum, which consists of lactic acid and fatty acid. Now we all know acid means pH will be less than 7, and acidic environment inhibits the growth of most microorganisms. So secretion of sebum is actually acting as anatomical barrier, right? Because they are resisting the growth of microorganisms due to their small pH, due to their less pH. Now, do you know what is acne? A few bacteria that metabolize sebum live as commensal on our skin, and sometimes they cause a severe form of acne. Now, normally Teenagers suffer from acne. Most of you, many of you must be suffering from acne. I've also suffered one time. Now, one very good or effective acne drug is there called isotretinoin, which is a vitamin A derivative. I don't know whether your dermatologist have ever prescribed you or not, but my dermatologist once prescribed it to me. That is isotretinoin. It's a vitamin A derivative that prevents the formation of sebum. Okay. Now, this is the second point that is written here. Acidic environment retards growth of microbes. Now, the first point written is mechanical barrier retards entry of microbes. What do we mean by that? See, until and unless there is a break or there is a cut or there is a wound on our skin, nothing can enter our skin from outside. No foreign agent can enter our skin. So in that way, skin acts as a mechanical barrier and retards the entry of microbes. Next comes the mucous membrane. Mucous membrane, we all know, is present in our alimentary tract, respiratory tract, urogenital tract. They all are lined by mucous membrane. Mucous membrane is not any dry protective layer, right? Mucous membrane means it makes secretion which is called mucus, which is a viscous fluid. And what is the function of this mucus? It entraps foreign microorganisms. Okay, it entraps foreign microorganisms. In the lower respiratory tract, it is covered by cilia, which is hair-like protrusion. Now, what is the role of cilia? Their synchronous movement propel the mucus entrapped microorganisms from this tract. Okay, if microorganisms enter, they get entrapped by mucus, and then cilia propel those microorganisms out of our body. So these all are part of non-specific host defense that is present from our birth. Okay, we are born with this kind of host defenses. So cilia propel microorganisms out of body. And first point that is mentioned here is normal flora compete with microbes for attachment site and nutrient. Now, what do we mean by that? Normally in our body, say for example, skin surface, some resident flora is always present. Like in our gut, E. coli, always present. They are known as resident flora. That means they are the resident of that particular part of our body. Now, what is the advantage of this normal flora? Because they have already occupied a certain place, so they will outcompete pathogens for attachment sites on the epithelial cell surface. And since they are already present, so they gather all the nutrients, so microbes won't get any kind of nutrient. So normal flora actually help us and they compete with microbes for attachment sites and nutrients. So this is about anatomical barrier. Next is physiological barrier. Now, what is physiologic barrier? First is temperature. Normal body temperature inhibits growth of some pathogens. This is true for some chicken, like for example, have innate immunity to anthrax because their high body temperature inhibit the growth of this anthrax bacilli, or what we call it Bacillus anthracis, right? So normal temperature inhibits because at normal temperature, what will happen? Pathogen, the body contains the protein and the protein will be denatured. Fever response inhibits growth of some pathogen. Same way, the pathogen's body protein will get denatured at high temperature, right? Then low pH. Acidity of stomach. We know that stomach contains gastric juice. The pH is almost 1 to 2. It's highly acidic, and the acidic environment kills ingested microorganisms because microorganisms are not able to tolerate very high acidic environmental condition. So they will obviously die. Then chemical mediators. Okay. Inside physiological barrier, few chemical mediators come. First is lysozyme. Now, lysozyme is present in different secretions of our body like saliva, then tear that is secreted from lacrimal gland. Now, what is the specialty of this enzyme? Lysozyme is such an enzyme which is antibacterial. How? Because they cleave bacterial cell wall. We know that bacterial cell wall contain peptidoglycan and lysozyme cleaves that peptidoglycan layer. Second is interferon. Interferon is a kind of glycoprotein which is secreted by virus and that induce antiviral state in nearby uninfected cells. We will describe this topic later on in our next video or sometimes later. Then complement. Complement is also a protein that lyses microorganism or facilitates phagocytosis. Complement is such a system which is there to complement the action of antibodies. Okay, that is why the name complement. There is a separate chapter on this topic. Then comes few toll-like receptors that recognize microbial molecules. Toll-like receptors that recognize microbial molecule. We call it TLR. Now TLRs recognize lipopolysaccharide found on gram-negative bacteria, and the mechanism for this response is via TLR on macrophage that recognize LPS and elicits a variety of molecules in inflammatory response upon exposure. The last one is colectin, which is a surfactant protein and that may kill certain bacteria directly by disrupting their lipid membranes. So these all come under the chemical mediators. Then comes phagocytic or endocytic barrier. Phagocytosis. We all are very much well acquainted with this term from our, I think, 8th or 9th standard. Phagocytosis, another name is cell eating. Phagocytosis is an important defense mechanism because it causes the ingestion of extracellular particulate material by this process. It is one type of endocytosis, a general term for uptake by a cell of material from the environment. In what happens in phagocytosis, a cell's plasma membrane expands around the particulate material, which may include the whole pathogenic microorganism to form large vesicle called phagosome. Most phagocytosis is conducted by specialized cell like blood monocyte, neutrophil, tissue macrophages, etc. Okay. Then finally is the inflammatory barrier. Inflammatory barrier, tissue damage and infection induce leakage of vascular fluid containing serum protein with antibacterial activity. After 2 or 3 slides, we will discuss the inflammatory barriers in a bit detail. Now, secretions at epithelial surface, I have just mentioned here. From eyes, lacrimal gland or tear, secretion contain lysozyme, IgA and IgG. From ears, sebaceous gland, as I have already mentioned, oily secretion sebum consisting of lactic acid and fatty acid. Then from mouth, it's saliva, salivary gland, digestive enzyme, lysozyme, IgA, IgG, lactoferrin. Then from skin, sweat gland, sebaceous gland, and stomach, gastric juice. Now, I have mentioned here a few important points. Pay attention, please. Gastrointestinal, respiratory, epithelia, and phagocytes throughout the body are also known to produce a number of small peptides, which have potent antibacterial properties. Okay, they are called peptide antibiotics. These peptides have molecular weight 3 to 5 kD. That means they are very small in size. And they include secropin, magainin, and defensin. Okay, secropin, magainin, and defensin. They are of very small molecular weight. Now they are the part of the body's innate defense mechanism and are highly conserved throughout species, probably representing one of the most primitive defense mechanisms against microbes. Okay, so secropin, magainin, and defensin. Although their mechanism of action are different, these peptides are effective against both gram-positive and gram-negative bacteria. Whereas secropin and magainin cause lysis, others interfere with ion transport. So these are their mode of action. Secretion is upregulated as a result of bacterial infection. Now, few non-pathogenic bacteria also exist in our body. As I have already mentioned, they are known as resident flora or normal commensal. They are found on the skin, in the mouth, and in the reproductive tract and gastrointestinal tract. Gastrointestinal tract contain many billions of bacteria, and they have a symbiotic relationship with us. Now, these bacteria help to prevent pathogen from colonizing the site by preventing attachment by competition, as I have mentioned, competition for space, for essential nutrients, and there is one more way by which they outcompete the invading organisms, and that is by releasing antibacterial substance, and that substance is called bacteriocin, as for example colicin, antibacterial protein. Now I just want to mention that my PhD work is on bacteriocin. If you guys are interested to know in detail about this bacteriocin and my PhD topic, just mention it in the comment section. I will definitely make a separate video on this particular topic. So what I was telling, that bacteria, that is normal resident flora, they outcompete the invading pathogen by producing bacteriocin. Bacteriocins are nothing but antibacterial peptides which inhibit closely related species, okay, induce a sequence of events, and those events are collectively known as inflammatory responses. The end result of inflammation may be marshalling of a specific immune response to invasion or clearance of the invader by components of innate immune system. There was a Roman physician, Celsus, who described inflammation has four cardinal signs. He called it as four cardinal signs of inflammation. And what are those signs? Number one is rubor. Rubor means redness. Number two is tumor. Tumor means swelling. Number three, calor. Calor means heat. And number four is dolor. Dolor means pain. Now, have you ever noticed when you suffer from some insect bite or some wound, then what happens actually? Yes, the particular region becomes red first, then it swells, then that particular area becomes hot and you suffer from pain. So now you can easily justify the four cardinal signs of inflammation: rubor, tumor, calor, and dolor. These are the events. And in 2nd century AD, another physician, Galen, added a fifth sign which is called functio laesa, or loss of function. Now, major events of an inflammatory response, first is vasodilation. Now, vasodilation is, as the name suggests, an increase in the diameter of blood vessel, okay, and which is responsible for tissue redness or erythema and increase in tissue temperature. Now, as the increase in capillary permeability happens, it facilitates an influx of fluid because the permeability of the capillary is increased. So obviously, from outside, fluids and cells will enter the tissue. The fluid that accumulates is called as exudate and has much higher protein content, and this accumulation of exudate from outside contributes to tissue swelling, or what we call as edema. Third is influx of phagocytes from the capillaries into the tissue. It is facilitated by increased permeability of capillaries. Now, the immigration of phagocyte includes the following steps. That is the influx of phagocyte from the capillary to the tissue. It occurs by following three steps. First step is margination. What is that? Adherence of cell to the endothelial wall of the blood vessel. Second is diapedesis or extravasation. That is immigration of capillary endothelial cell into tissue. And number three is chemotaxis, their migration through tissue to the site of invasion. Now, this was all about innate immunity. Now, let's start with adaptive immunity. We will discuss much less about adaptive immunity because already we have mentioned the most important points of adaptive immunity. Now, adaptive immunity displays four characteristic attributes. You must memorize these four attributes, okay? First is antigenic specificity. Number two is diversity. Number three, immunologic memory. Number four, self, non-self recognition. We will discuss all these points later on. Now, adaptive immunity, as I have already mentioned, is not independent of innate immunity. Through the carefully regulated interplay of adaptive and innate immunity, the two systems work together to eliminate a foreign invader. So this is all for this video. Thank you for watching. Stay tuned for the next part. All the best.